In Chapter 3, we're going to continue our discussion with transcription and RNA processing. So at this point in time, we've talked about the ability of DNA to be able to duplicate itself. Now we're going to talk about how DNA makes an RNA copy that will eventually get translated into protein. So let's first overview transcription. Let's talk about a couple of key points, some facts about transcription. First of all, not all the DNA in your genome is going to be transcribed. Of the 3 billion base pairs of DNA that you have, only 3 to 4% is actually transcribed into mRNA, ribosomal RNA, transfer RNA, or small nuclear RNA. In other words, most of the DNA in your genome contains information that's either promoter sequence, enhancer sequence, spacer DNA, telomere DNA. It's mostly DNA that does not get conveyed into protein information. So what does it do? We'll leave that to the students who take the test 20 years from now. RNA polymerase, another key point, is that it initially finds and binds to the promoter, not to the coding region. So when the polymerase comes in and looks for a spot to begin transcription, it's not looking for coding region DNA, it's actually looking for promoter region DNA. Another fact is that what we refer to as the gene is actually double-stranded DNA, but only one of those strands of DNA is going to be used as a template for transcribing into messenger RNA, transfer RNA, ribosomal RNA, what have you. So a gene is double-stranded DNA sequence, but only one of those strands is used as a template for transcription. What does that mean then? Well, there's a template strand that's used for transcription, and it's going to be complementary and anti-parallel to the mRNA that gets synthesized. The other strand, therefore, we call it the coding strand, it doesn't get used. It just sits there, it looks pretty, it ends up being identical to the messenger RNA with a couple key exceptions. Whereas there are T's in the coding strand, you will see U's in the mRNA, because remember, there's no U's in DNA, and there's no T's in RNA. The coding strand is also going to be the strand of DNA where you see the TATA box or the CAT box. Complementary to that, on the template strand, you're going to see a GTTA box where you saw the CAT box, and you're going to see a AT on the template strand where there's a TATA box on the coding strand. So when we refer to things like the CAT box or the TATA box, we're referring to the sequences as they read on the coding strand, not the template strand. Shown on this particular figure is a schematic of a typical transcriptional apparatus. Notice, DNA is always read from 3 to 5 prime direction, and that the RNA is always going to be synthesized 5 to 3. Those rules have not changed. They're the same rules that applied for replication. They're the same rules that now apply for transcription. So the DNA template is read from 3 to 5 prime. The RNA in this case is synthesized in the 5 to 3 prime direction. So if you look at gene 1 right here, you'll see that transcription, the arrow is telling us that it goes from left to right. On the real exam, they might not tell you which direction the transcription's going in. They might tell you that there's a promoter sequence at this area and a termination sequence down here. And you're expected to understand that transcription goes from a promoter towards the termination sequence. So that information alone would allow you to deduce that transcription is going to occur from left to right. Okay, so having that information, tell me which is the coding versus the template strand. We have a top strand and a bottom strand. So which is the coding strand? It's going to be the top strand. Now, how did you know that? The reason you should have known that is because the template strand is always going to be read from 3' to 5'. We said that transcription is happening from left to right. So in order to be read from 3' to 5' from left to right, that makes the bottom strand the template strand. It's the only strand that goes from 3' to 5' in the left to right direction, thus making the other strand the coding strand, which would then go from the 5' to 3' orientation with respect to the transcription. Let's flip over here to gene number 3. In gene number 3, which direction is transcription happening? It's happening from right to left. And why do we know it's happening from right to left? It's because it goes from the promoter to the termination sequence. anytime you see it going from the promoter to the termination sequence, that's the direction that transcription happens in. The second question then is, which strand, in this case, we'll ask, which strand is the template strand? Well, the template strand, remember, has to go from 3 to 5 prime in the same direction as the transcription is occurring. So in this case, 3 to 5 prime is on the top strand, and that's the same direction as transcription. So this strand would be read from 3 prime to 5 prime by the polymerase, and what would be synthesized is complementary RNA in the 5 to 3 prime direction. So yes, for this particular gene 3, the template strand is actually the top strand, whereas the coding strand is the bottom strand. So I think this illustrates for you that just because the top strand was the coding strand for gene 1 doesn't mean that the top strand has to be the coding strand for gene 3. Gene 3, it's actually the bottom strand that's going to be the coding strand, whereas the top strand was the template. So make sure you understand that although a strand of DNA might be the template or the coding for one particular gene, that doesn't make it the template or coding strand for every gene on that chromosome. It just depends on the orientation of the promoter with respect to the termination sequence. So what are the types of RNA produced within eukaryotic cells? They're going to be the following. They're going to be messenger RNA, ribosomal RNA, transfer RNA, and snRNA, which stands for small nuclear RNA. Which of these is the most abundant RNA species in the body? Believe it or not, it's ribosomal RNA. Okay, ribosomal RNA is the most abundant RNA species in the body. It's not messenger RNA. A question is the promoter transcribed? Is this promoter region that you see here going to be transcribed? And the answer is unequivocally no. The promoter region is never transcribed. So if you're given a question on test day where they have some DNA sequence set up and you're asked, what would the messenger RNA transcribed from this look like? Never choose the answer that has promoter sequence included in the answer. The promoter sequence is never transcribed. That's a trick question. And so therefore, no, the promoter is not transcribed. The position where transcription starts, we call it the +1 site. That's where transcription is going to begin at a site called the +1.